Today, we're going to talk about the Ames test. The Ames test uses bacteria to test different compounds for their mutability or their potential to cause cancer. We use bacteria because bacteria have large population sizes, and they will conveniently form individual colonies, which consist of clones. So let's take a moment to talk about this. So if we have a flask of 500 mils of bacterial culture, and we take some of this and we put it on a petri dish with agar, then we're probably going to end up getting a lot of colonies. Maybe we dilute it so the colonies are fewer. But how do we actually get these individual colonies? Well, each one of these individual colonies was started by a single cell. So in this spot on the plate, where I just pointed to, we had one single cell that was from this culture liquid. Again, diluting is usually necessary. And that single cell is not going to stay a single cell. It's going to divide, and that's going to divide. And then we're going to get lots and lots of bacteria. And there's really not much potential for transduction here. There could be, I guess, conjugation, but it would be between these bacteria that are all very closely related. And so you're going to get what is basically a colony. Should have done this in yellow, but I didn't. A colony that was started by a single cell and which consists of clones of one another. In other words, they have identical genomes. But just because we have this one cell or this one colony, which consists of a bunch of clones, it doesn't mean that it is identical to this colony. Because this colony was started by a different cell. And that cell might actually be different than our cell number 1A. We'll say this is 1B. 1B might have a different genome. It might have some kind of genetic difference that's distinct from 1A. So the idea here is to have our bacterial liquid culture. And our bacterial liquid culture has a specific strain of bacteria. This strain of bacteria cannot make a specific amino acid, which is histidine. So there's some kind of mutation. It's a small mutation. I think that it's a SNP. You can watch my other video on types of mutations. So this is a single nucleotide polymorphism. Again, very small. It's a single base pair that was changed, which means that this whole set of genes or this whole operon for making histidine doesn't work in this one strain. And so they will only grow on plates that have histidine provided in the media. So that would be His plus. And we would get bacteria growing up on here. If we did that whole process again, we would get colonies on the His plus media. And then in the His minus media, we would get nothing because the bacteria can't produce histidine, and you need histidine to make proteins. So we wouldn't get anything there. Okay. So then the step that actually tests for whether or not something is a mutagen is we expose this culture That's the second time I've done that today. We expose this culture to the mutagen. So let's just say maybe it's UV light, even though we already know that that is a mutagen. So we expose the culture to UV light and we plate it to see if bacteria grow on the histidine plus media, which they should. This would be the control. And then we also observe whether or not bacterial colonies show up on the histidine minus. What can we conclude if we do see a colony that shows up on the histidine minus? Well, we know now that this bacterium can produce histidine, which means that it gained a mutation. And again, mutation can just mean a change in genetic sequence. So it's genetically different from these bacteria that cannot make histidine just because of a single SNP, because it fixed that SNP, in other words. It had a mutation which allowed this genetic pathway for making histidine to be fixed and be restored. And what we want to do then is compare the number of colonies or the number of instances where bacteria are mutated with the number of colonies where bacteria are not mutated on the Hist plus. And then we'll see the mutability of whatever molecule or condition we have here, which we're testing. And of course, we also need to isolate these colonies. We need to study them. We need to look at this and see what the genetic difference is, and if this is heritable, if this is just a fluke. But this is how we still go forward in testing new compounds, initially at least, to see if they might be of danger to humans. So again, that's the Ames test for mutagenicity.